go.
Alright, so this is a little chunk of DNA, and we're going to model how it replicates itself. Remember that DNA needs to be replicated before a cell can divide, so that both new cells have all the DNA or all the instructions to make proteins for that specific cell. Because the DNA bases always pair with each other, thymine with adenine and cytosine with guanine, I said that backwards, but yes, that's what makes it really easy to replicate. So when your body is ready to replicate its DNA, it sends in an enzyme. It's called helicase. You don't need to memorize that, but that's what it's called. An enzyme comes in and it breaks the DNA apart. Remember, these are pretty weak bonds in between the base pairs, and that's why it's able to be broken apart. So it pulls the DNA apart. You can imagine the DNA continuing on and on and on up ahead. Pulls the DNA apart, and then a second enzyme comes in and pulls free nucleotides that are just hanging out in the body because from the food we eat, like bananas, that contain DNA. So we take these free nucleotides and they just match up. So this is the second enzyme. Its name is polymerase. Let's see, we've got a thymine over here, so your body would match that with an adenine, guanine with a cytosine, adenine with thymine, guanine. It's kind of laborious, but nope, that does not match. But now you know how your body does it. No, my cytosine short. Guanine. Yeah, so now you have one DNA strand that's the same as the first, and that at the same time it's rebuilding this one as well. This is enzymes doing the work. This one's name is polymerase. Ta-da! And so now, from one DNA strand, you have two DNA strands that are exactly identical to each other and exactly identical to the original DNA strand. So that's how your body replicates DNA. So just to recap, your body breaks the DNA open on the hydrogen bonds in between the nitrogen bases with one enzyme, and then the second enzyme comes in and adds free bases to the half strands, making them both into complete strands. Again,